Hello everyone, this is Hasiba Asma. I'm a PhD student in Dr. Mark Halfon's lab in State University of New York at Buffalo. Welcome to my poster that's about improved enhancer discovery in Drosophila melanogaster and other insects. So enhancer is a short stretch of DNA where a number of transcription factors can bind and control the spatiotemporal expression of nearby genes. As you can see here, these enhancers are being represented by these O-ring bands, and these are bound by small transcription factors, and that's how it's regulating the expression of this nearby gene. Thus, their identification is very important for understanding the gene regulatory network. We have previously developed an effective computational method for genome-wide enhancer prediction. We call it SCRMSHA. That stands for Supervised Cis Regulatory Module Prediction Program. This is how the basic framework of SCRMSHA looks like. It uses training set that we extract from that is composed of known enhancers that we extract from RedFly database, which is comprehensive database of Drosophila melanogaster and has almost 25,000 experimentally validated enhancers in it that are obtained from published literature. This was also developed by our lab. So the trained model is then used to predict enhancers in not only Drosophila but in a wide range of species, holometabolic species such as mosquitoes, beetles, and bees. And SCRMSHA has proven to be quite effective. In fact, we have had 75% success rate to discover enhancers in cross-species fashion in genome as far diverged as 345 million years ago when tested empirically using reporter genes in these transgenic animals. So we are currently working on different approaches to improve effectiveness of our SCRMSHA method to even more. Our first approach, which is toward individual prediction confidence score, we use comparative genomics in order to give confidence score to each of our SCRMSHA method prediction. So for this, our assumption is that if SCRMSHA prediction is a real CRM or enhancer, then we should be able to predict it in multiple related species, and that should be within orthologous locus, as can be seen here. So incorporating such confidence score to SCRMSHA's output will be a powerful improvement, as it will provide a way to discriminate between true positive and false positive by giving high confidence score to true prediction than to false prediction. Then our other approach is improving SCRMSHA's performance through iterative search and validation strategy. So the basic idea is to train SCRMSHA, get the prediction, experimentally validate some of those predictions, and based on the result, update the training set and use that training set, the updated training set, to get the predictions again, and that with the improved performance, and redo the predictions, and that will be with the improved performance. So to identify those enhancers that are potentially involved in abdominal pigmentation, we first used our SCRMSHA on a small training set of just 7 enhancers that drove gene expression in the adult abdomen as a first round. So once we got the prediction from SCRMSHA, we have done empirical testing of 18 of those predictions. And empirical testing revealed 10 new enhancers with the same activity. For example, this KRH1 was one of those predictions that turned out to be true positive, and this PDB1 was among 8 false positives. So we took those 10 new enhancers that were true positive, or that drove the correct expression pattern, and combined them with the original 7 members of our training set to generate a new expanded training set for running a second round of SCRMSHA. When we analyzed the result, our predictions from second round of SCRMSHA, we found that top prediction result from this round did not include 7 of the 8 sequences previously found to be false positive. But it still continued to it still contained all the 10 of the previously given true positives. And we also did some empirical validation. And we actually did an empirical validation of 21 of those predictions from the second round. We found that out of 21 enhancers, we had 95% were functional enhancers. And among those, 81% had the expected expression pattern. So these results clearly demonstrate that iterative approaches can serve to augment the weak training set to improve our true positive to false positive ratio.